Well, good morning, everyone. So my name is Katie Resvani. I'm a stem cell transplant physician and cell therapist at MD Anderson Cancer Center. Of course, Professor Max Torp is from Würzburg University. It's an honor to be here today. And really, it's an honor and pleasure today to introduce Professor Chiara Bonini. I don't think she needs introduction to many of you. She's a pioneer in the field of TCR-engineered T-cells and CRISPR gene editing. She trained in Milan, in New York, and in Seattle. Since 2008, Professor Bonini's been the head of the experimental hematology unit at San Raffaele University. She's also Deputy Director of Immunology, Infectious Disease and Transplantation. She's received too many awards for me to really mention. She's the recipient of multiple recognition. She's on multiple patents and over 130 different publications. So it really is, I think, an honor for us to hear her talk on CRISPR gene-edited technologies today.
Took the title as we decided it at the beginning, but I will give you a spoiler. We're going to talk about T-cell therapy for cancer and how we can exploit genome editing for the purpose. Now, I think we can all start from a good premise, which is that the T-cells have the potential really to cure cancer, and they've demonstrated it clearly in the context of allogenic stem cell transplantation, where donor T-cells infused with the graft can recognize and kill cancer cells at a price of toxicity such as graft versus host disease. We've seen it also in the context of autologous setting with tumor-infiltrating lymphocytes, and more recently with CAR T-cells that proved highly effective in selected B-cell tumors. Now, saying that, how can we export this experience, this starting point, to treat other cancer types? There are a number of challenges that were very nicely identified by Wendell and June in this review. And we've tried in years in the lab in San Raffaele to address them with different projects. We've been working at increasing the safety profile of T-cells, or try to increase their ability to persist by manufacturing protocols that allow the generation of early memory T-cells, such as memory stem T-cells or central memory cells. What I'm going to focus on today is these two challenges. So how should we redirect T-cell specificity, and how should we try to remodel the tumor microenvironment, which is usually an immunosuppressive microenvironment? And I'm going to focus on these two because these two really requires a genome editing approach and would much benefit of a genome editing approach as opposed to a conventional gene transfer approach. So let's see if it works. It's next. Okay. So what I'd like to try to convince you in this presentation is the fact that we can redirect T-cells not only by introducing a CAR, so a chimeric antigen receptor, but also by exploiting what is the natural mechanism by which T-cells recognize their antigen, which is a TCR, T-cell receptor. So we can identify theoretically TCR from tumor-redirected, tumor-specific lymphocytes, and use the gene and introduce them in healthy T-cells of other subjects, other patients. And why should we do that? What could be the advantage of a TCR approach compared to a CAR? There are a number of reasons. I think the most important one is the mechanism by which a TCR works. It does recognize peptides processed and presented on MHC molecules. This means that as opposed to CARs, a TCR can target essentially every protein that is present and expressed in the cancer cells, independently of its location, while CARs only see native proteins present on the surface of cancer cells. So we had a much wider basket of antigen from which we could pick our target. Another potential advantage is physiological activation. TCR is the natural protein that provides signal to T cells, so not only signals that activate T cells, but also that provide a signal for survival, for proliferation. And I'm not sure that with CARs we reached exactly the same wideness of effects. And finally, preliminary data, but I would be more careful in this since we have much less patients treated with TCR-redirected T cells than with CARs, but preliminary data suggests a better safety profile for TCR-redirected T cells than for CAR T cells. Really, I need to find a way to make this work. So, if we want to redirect T cells with the TCR, first thing we need to do is to select an antigen, and there are a number of characteristics that this antigen needs to have. It needs to be expressed by many cancer cells, specifically expressed by cancer cells, and not expressed or expressed at very low levels by normal cells. It needs to be possibly relevant for oncogenicity and for tumor progression. And the fact of being able to target an antigen that is present on inside the cells and not on the cell surface, it's a major plus. Because we want to try to target something relevant for the cancer cells so that it will be difficult to evade the immune response that we are eliciting or producing. So looking for such an antigen, we started taking advantage of a prioritization activity performed by the NCI many years ago and ended up targeting WT1. This is an antigen expressed by essentially all acute myeloid leukemia, but also by some other tumors. It's relevant for oncogenicity. It's expressed also by relapsing blasts. And it is immunogenic. Vaccination trials and trials with T cells, natural or TCR redirected T cells have already proved that this antigen is an antigen with a good safety profile. So we wanted to use a TCR gene editing approach to target WT1. The first thing we needed was to identify TCR specific for this antigen. And Eliana Ruggero in the lab, a project leader in the lab, set up a pipeline to identify TCR specific for different antigens, starting from healthy donors or from tumor tissues. And I'm going to drive you through this pipeline with an example, which is at the end the TCR that we selected among the 19 TCR that we identified from healthy donors and specific for WT1. So she started from 15 healthy donors, and she paused PBNC of these donors with overlapping peptides encompassing the entire WT1 protein, and looked at the response after different cycles of simulation. And as you see, in 14 out of 15 donors, she was able to see a response, sometimes early as S4, in other cases a little bit later during the simulation. But in 14 out of 15, a response was there. It was measured in terms of interferon expression or degranulation by different approaches. And it was also proved by the fact that the cultures that were highly polyclonal at the beginning started to become monoclonal. So there always were a dominant TCR present in the culture in parallel with the recognition. Moving from this stage, we decided to try to identify the peptide that was recognized. And an interesting observation that we made was that is probably due to the fact that the majority of the donors from which we started were typed HLA-A0201, and we choose this because this is a very frequent HLA, and we want to try to have a TCR exploitable in a high number of patients, so restricted by a common HLA. So what we observed was that there were some immunodominant responses. So these are different donors, all responding to the same subpools of WT1 peptides. So we got interested in this response, and we used the deconvoluting grid to, I think we have a problem with the battery here. to identify the exact peptide present in the subpool. And every peptide was present only in 2 subpools, so we could predict the peptide that was recognized in this, by this immunodominant response. Of course, then the prediction was experimentally validated. And then we ended up, oh, thank you so much. And then we ended up really characterizing the different TCRs. So what did we do? We looked at in silico prediction to identify what was the HLA molecule present in that specific donor, and that was the restriction element for the peptide. In this case, we predicted HLA-2. Then that was experimentally validated by pulsing the pool of WT1 peptides on an HLA-2 cell line, and also pulsing the specific peptide on the same cell line. And the final validation we made before deciding that TCR was really a relevant tool for TCR gene editing was looking at its processing. So we really wanted to ensure that cell lines, or in many cases AML blasts taken from patients, were able to process that specific peptide, and so were recognized by our culture. Seeing this, we got interested in the TCR genes. So all this work was done with the cells, with the natural cells present in healthy donors. So from there on, we started being interested on the TCR genes. And we identified the TCR genes by longitudinally tracking by TCR sequencing the alpha and beta TCR genes in the different cultures. You see, in some cases, it was really easy to identify and couple the alpha and beta chains. In other cases, it was more difficult, but the longitudinal tracking really allowed to identify it in all the donors. And in some donors, we had more than one TCR specific for WT1 epitope. So with this approach, we ended up, as I said before, with 19 different WT1 specific TCR. We got mostly interested in the CD8 so HLA class 1 restricted TCR, and much more interested in the A2 restricted TCR, which are specific actually for 2 different peptides. We ended up selecting the TCR specific for the VLD. So once we have our library, and we published this library and focused on the VLD peptide in parallel with the group of Phil Greenberg, who from a completely different project reached the same peptide as a relevant one. Having this peptide, this TCR ready, having a number of TCR ready, and we need to find a way to choose a strategy by which we can express this TCR into healthy T cells. So the easiest way to do this is to build a lentiviral vector or a retroviral vector encoding for a TCR alpha and beta gene of the WT1 specific TCR and transduce healthy T cells. But by doing this, we will end up with cellular products which are not really identical to the natural counterpart. They do express our tumor-specific TCR, but also the endogenous repertoire. So there is a mutual dilution of the TCR, reduced expression of the TCR, reduce avidity of the T-cell product, and reduce efficacy at the end. And finally, we might have mispairing between the endogenous and tumor-specific TCR chains, leading to new unpredicted specificity, potentially autoreactive. So to overcome these limitations, several years ago, with Luigi Naldini and his team, we decided to use genome editing. It was before even CRISPR were available, so we focused initially on zinc finger nucleases. But the point was that no matter if we use zinc finger or CRISPR as today, or TALEN or other nucleases that are available, the point is that with this technology, we can not only add a gene to the cell, but we can actually also disrupt a gene. So eliminate a gene on the cells, because the way by which these tools work is to create DNA double-strand break in an area, in a genomic area of DNA that the researchers select, and by doing this, the cell, to survive, will have to cope with it. And it will cope mainly by non-homologous unjoining, which is a quick and dirty approach in which indels are introduced. And if these are introduced in a coding genomic area, we will lose the frame and have a gene disruption. Another way by which a cell can cope with the break is homology-directed repair, which is a much more sophisticated approach that we can exploit to enforce and induce a targeted integration of a cassette of DNA that we selected in origin, for example, encoding a tumor-specific TCR, and express it and integrate it exactly in the same gene that we want to disrupt. So by this approach, we're going to move from gene addition, which is what you can obtain with the gene transfer vector, to substitution of genes, substitution of function. So the TCR gene editing works in this way. So we can use zinc finger nucleases or CRISPR-Cas9 directed to the constant region of the endogenous TCR repertoire to disrupt the TCR genes, have naked T-cells, and then we can integrate in the track or add by a lentiviral vector the tumor-specific TCR. So now these products will express only the TCR that we have selected. As I said, we started this project several years ago and showed that TCR-edited T-cells are superior to TCR transfer T-cells. But the protocol with zinc finger nucleases was really very complex, too complex for clinical translation. So 4 genetic manipulation, 45 days of culture was really a bit of a nightmare. So fortunately, the CRISPR revolution came and we could exploit CRISPR. And thanks to a collaboration with the Intellia Therapeutics, we set up much simpler protocols. These are the 2 protocols we compared, one with lenti transferring the tumor-specific TCR in T-cells in which we have simultaneously disrupted TRAC and TRBC, and the second one is based on the targeted integration. So in one single gene modification moment, we disrupt TRBC and force targeted integration in TRAC of our cassette and end up with our tumor-specific TCR. Both approaches are very efficient. The level of TCR disruption is really, really high with CRISPR. The expression of the tumor-specific TCR is also high. And most importantly, both protocols can be applied in conditions that allow the generation of cell products with a central memory in orange or stem memory in red a phenotype, which we've seen. It's a very important phenotype for persistence of T-cells, of engineered T-cells in humans. So since this story has been published very recently, I will not show you all the comparison of the different TCR on the edited cells. I will just show you how it ended up with the TCR that we selected. We proved it able to control a primary blast from HLA-A2 patients and grafted in NSG mice nicely, and also some cell lines. So with this TCR, we ended up starting a clinical trial. So what I've shown you with this part of the project up to now is how we started from 14 donors, ended up identifying tools, and then at each step of validation, we sorted out some of the low priority TCR and ended up with a protocol validated, a TCR validated, ready for a trial that has started at the end of 2021. With this, I'll end up thanking all the team, all our collaborators, Fabio Ciceri, I didn't mention Monica Casucci, head of the CAR team in San Raffaele, and all of you for your attention. And I'll be glad to take questions.